Good afternoon, everyone. So, today we will have the seventh tutorial session, which basically focuses on looks at the basics of immune system and immunology, and we will then look at into few questions. So, maybe it's 3-7 now. We will wait for 3 more minutes, 3 to 5 minutes, and then we will start in case others want to join. Okay. Yeah. So we will start the session. I will start sharing the screen. So immune system basically is a complex group of defense response which are found in organisms that helps repel or fight against disease causing pathogens. And immunity from a disease is actually uh, a result uh, or is actually affected by two cooperative defense systems. One is the non-specific innate immunity, and one is the specific acquired, which is also called as acquired or adaptive immunity. So, very briefly, non-specific protective mechanisms actually help in repelling all the microbes equally, whereas the specific immune response are tailored to particular types of pathogens. Okay. However, both innate as well as adaptive immunity systems work together to thwart organisms from entering and basically to prevent organisms from entering and proliferating within the body. So, these immune mechanisms also help in eliminating the abnormal cells of the body that can develop into cancer. So, very briefly, the first line of defense or the barriers are one is the skin, which is one of the major barriers which stops germs from entering our bodies. And even if the germs can get inside through nose or mouth, then we have the next line of barriers, the immune system and other cells and stuff to fight them. Second line of defense or general attackers are phagocytes. These are special white blood cells that act like scouts, and these cells actually find and destroy the invading pathogens. And there's inflammation and fever. Whenever in general our bodies are attacked by any pathogen, it can trigger responses like inflammation and fever. Fever, for example, can make it harder for some bacteria to grow. The higher temperatures, is actually a defense mechanism by our bodies to prevent some pathogenic bacteria to grow inside our bodies. And so overall, and this is something that we always are born with. Like we don't have to specially produce this inside our cells and stuff. And however, I mean, we don't get it from elsewhere. These are already present in our body by birth, and we don't. But however, these things don't create lasting protection. Okay. The lasting protections are actually given by the third line of defense, which are specific attackers, and eventually these produce memory cells as well, memory immune cells. So one is lymphocytes, which is another type of white blood cell. Whereas some lymphocytes are there, which are called B cells. These produce antibodies, and antibodies are like targeted missiles, right? That attach to specific pathogens or germs, thereby marking them for destruction. And then there are memory cells, and after fighting off a pathogen, some lymphocytes become memory cells, and these cells eventually remember that specific pathogen. So even if it tries to invade the body in future, the immune system can actually fight it off much faster and more effectively. So this is eventually called acquired immunity. Okay. Now, if we talk about the external barriers to infection, the skin and the mucous membrane linings of the respiratory, gastrointestinal, and genitourinary tracts provide the first line of defense against invasion by microbes or parasites. So if we talk about skin, it has human skin has a tough outer layer of cells that produce keratin, and this layer is constantly renewed from below and serves as a mechanical barrier to infection. In addition, there are glands in the skin which secrete oily substances, including fatty acids like oleic acid, that can kill some bacteria. There are skin glands which can also secrete lysozyme, an enzyme to prevent tears, which is also present in our saliva and tears, which can actually break down the outer wall of certain bacteria. Even in the previous tutorial sessions, we looked at uh, plasmid DNA isolation and stuff like that. So there also, if you remember, the bacterial cells that we were targeting, I mean, if we had to purify plasmid DNA from bacterial cells, then we have to first break the cells open, right? So for breaking the cells open, we were treating the cells with something called lysozyme enzyme so that it can, degrade the cell wall of the bacteria from which we are trying to purify plasmid DNA. So similarly, this lysozyme enzyme is actually also present in our tears and saliva, which helps in fighting against some bacterial pathogens. Victims of severe burns can often fall prey to infections from normally harmless bacteria, which again is an evidence to the fact that intact, healthy skin is actually necessary for a healthy immune system. Now, when we talk about mucous membranes, like the outer layer of the skin, but these membranes are very much softer. It lines the respiratory, gastrointestinal, and genitourinary tracts and provides a mechanical barrier for cells that are constantly renewed. And a mechanical barrier, these cells, the cells that line these cells are constantly renewed. The lining of the respiratory tract has cells that secrete mucus, which is also called as phlegm, which traps small particles. There are other cells in the walls of respiratory tract which have small hair-like projections called cilia, which steadily beat in a sweeping movement, thereby propelling the mucus. And in case any particles are trapped in the throat or nose, it can eventually get removed along with the mucus. So also present in the mucus, actually in the mucus, we do have protective antibodies as well, which are products of specific immunity. Cells which are lining the gastrointestinal tract also secrete mucus. Definitely the mucus helps in, because our stomach secretes hydrochloric acid. It has a very low pH. So mucus layer actually helps in preventing our stomach walls uh, from degradation by the acidic pH. However, in addition to passage of food, also mucus layer helps in smooth passage of food. However, mucus secretion inside our gastrointestinal tract also helps in trapping potentially harmful particles and preventing them from attaching to cells that make up or the cells that line the gut. Protective antibodies are secreted by cells underlying the gastrointestinal lining, and eventually the stomach lining also secretes HCl, which is strong enough to kill many microbes. Okay. Mm. And then there are chemicals which are involved in normal body processes but are not directly involved in defending the body against any pathogenic attack or diseases. However, they do help in kind of repelling the pathogens. How? So there are chemicals that can inhibit the potentially damaging digestive enzymes released from the body cells which have died in the natural course of events, and these can also inhibit similar enzymes produced by bacteria, hence limiting bacterial growth. There's another substance that provides protection against microbes incidentally to its cellular role. There is a blood protein called transferrin. So its normal function is to bind molecules of iron that are absorbed in the bloodstream through the gut and to deliver the iron to the cells. And uh, whereas we know that iron is required in general for cellular metabolism and stuff. However, the protective benefit transferrin confers results from the fact that bacteria like cells need free iron to grow. But when iron is actually bound to transferrin, the iron is unavailable to the invading germs or invading bacterial cells, and their growth is stopped. Then there are antimicrobial proteins as well, which form the complement system also. So a number of proteins contribute directly to the body's non-specific defense system by helping to destroy invading microbes. One group of such proteins is the complement system, which works with other defense mechanisms of the body, complementing their efforts to eradicate or fight the pathogens. So many microbes can actually activate complement in ways that do not involve specific immunity. However, once this complement system is activated, complement proteins work together to lyse or break apart harmful infectious organisms that do not have protective coats. And there are other microbes which can invade these mechanisms but fall prey to scavenger cells, which are again part of the immune system, which engulf and destroy infectious agents and to the mechanisms of the specific immune response. So complement system basically cooperates with both non-specific and specific defense systems in our bodies. Then there are interferons as well, which is another group of proteins that provide protection and inhibit the replication of many viruses. And cells that have been infected with the virus produce interferon, which eventually sends a signal to other cells of the body to resist the viral growth. It was actually discovered first in 1957, interferon at that time was thought to be a single substance. But since its discovery, several types of interferons have been discovered, and each type is produced by a different type of cell. So there are like alpha interferons, which are produced by white blood cells. Other than lymphocytes, there are beta interferons produced by fibroblasts, and gamma interferons produced by natural killer cells and cytotoxic T lymphocytes, or in general, killer T cells. So all these cells are also part of the immune system machinery. We will look at it one by one eventually. So all the interferons inhibit viral replication by interfering with the transcription of the viral nucleic acid. And interferons can actually exert additional inhibitory effects by regulating the extent to which these lymphocytes and other cells can express certain important molecules on their surface membranes. Then there are also proteins from naturally occurring bacteria which are naturally found in our bodies. So in the small and in our gut especially, there are uh, already gut loving bacterial species that are present, which are really important for our system to function. Okay. However, in those areas, the growth of invading pathogens is inhibited by these naturally residing gut bacteria in our bodies. And however, these do not cause diseases. But sometimes what also happens in case of gut bacteria is if it increases beyond a certain point, then the concentration increases beyond a certain point, then it can be harmful. Like suppose E. coli. E. coli, we do have a good amount of E. coli present in our bodies, which is actually very friendly and helps in digestion and stuff. Okay. However, if it increases beyond a certain point, then it can be really harmful. And also, E. coli bacterium is one of the top five leading pathogens in the world. Okay, like as in the bacterium causing pathogenic diseases like diarrhea and stuff. Okay, and yeah, so in general, these when in a proper optimum concentration, these actually are very essential for our body to function. So these bacterial species, the naturally present bacterial species inside the gut, can also secrete certain chemicals or variety of proteins that can um, enhance their own survival but inhibit the growth of any invading pathogen. Okay. Now, when we talk about the innate immune response, if pathogens pass the physical and chemical barriers, they eventually encounter the cells that destroy foreign substances or foreign attackers. So, these cells are non-specific effector cells of the immune system. And these non-specific effector cells include scavenger cells, which can attack the pathogens directly, and natural killer cells or NK cells, which destroy the infected or abnormal body cells. And then there are some immune cells which can eliminate pathogens through phagocytosis. Phagocytosis is like engulfing and digesting, eating up the pathogenic entity, and by engulfing it and digesting them. And then there are innate immune cells that interact with the acquired immune system to fight infections. So, if we talk about the scavenger cells which attack the pathogens directly, these are found in most animals and mainly consists of leukocytes or white blood cells. It was actually first identified by Mechnikov in 1884. And again, scavenger cells are further divided into two types. One is macrophages based on the sizes, little eaters, which are also called as granulocytes. And there are macrophages, which are really big in size, and also called in general micro and macro, as the name suggests, means little and big, respectively. And phages are phage, like phagy in general means eating. Okay, so macrophages, little eaters, macrophages, big eaters. Now, if we look at microphages, which are also called granulocytes, these are also called as polymorphonuclear leukocytes because they have a irregular nuclear structure. And these granulocytes contain granules with digestive enzymes and bacteria killing proteins. Again, granulocytes are of three types: neutrophils, eosinophils, and basophils. Neutrophils are the most common, which constitute about 60 to 70% of white blood cells. Neutrophils can engulf and destroy bacterial pathogens. Then there are eosinophil cells, which are effective against large parasites. And there are basophils, which release histamine and heparin, anti-inflammatory substances. These are these are actually important in allergic reactions. Okay. Then there are mast cells, which are similar to basophils and also help in immune responses. And granulocytes are actually produced in the bone marrow and can live only for a few days. And whenever there is any attack, they can move towards the infection site of infection via chemotaxis. Taxis means movement, and chemo means as in the movement due to any chemical signal. And there are some pathogens which can evade granulocytes using toxins or resistance to digestions. So then eventually our body needs help from specific immune system. Now, if we talk about first one was macrophages or granulocytes. Then comes macrophages, which are little bigger, and these develop from monocytes produced in the bone marrow. So monocytes circulate in the blood and then eventually differentiate into macrophages in tissues. And macrophages are commonly found in lymph nodes, spleen, and liver, which filter microbes. Macrophages are um, like macrophages have a longer shelf life than granulocytes. Macrophages can move more slowly and arrive later at the site of infection. Macrophages also engulf and digest pathogens, although it is comparatively slower than how granulocytes or macrophages um, phagocytose. And macrophages are important for activating specific adaptive immune responses. Okay, so in general, if we look here, so these are macrophages extensions. So macrophage is moving towards the bacteria. These green ones are bacteria. Okay, so macrophage eventually is moving towards the bacterial pathogen, and eventually the bacteria gets engulfed or phagocytosed inside the macrophage. Okay, this is here. Next comes bacterial pathogen contained inside a vesicle. These are again then trapped inside a vesicle here. Then lysosome fusing with the vesicle and releasing phagocytic enzymes. Eventually, lysosomes, these are suicidal bags, which the lumen of which contain like all the digestive enzymes at really low pH. So this is like lysosome, right? And this is the vesicle inside which the bacterial pathogen is trapped. So once the lysosome fuses here, uh, in this scenario, this is phagocytosed bacterial pathogen, right? And when lysosome is fusing there, it is also called as phagolysosome. Okay. So once the the lysosome fuses with the vesicle containing the bacterial pathogen, eventually phagocytic enzymes are released and the bacterial pathogen is destroyed here, as you can see, and digested. And then there are fragments, right? of or the remains of the bacterial pathogen which were undigested. So eventually these are thrown out or diffused out or excreted out of the system, as in secreted out of the system. So if we look, this is the scanning electron micrograph of a macrophage, this one, this pink or purple one, whatever you call it, and this yellow one is the uh, cancer cell. Macrophages can attack cancer cells as well. Okay. Then there are natural killer cells. So we looked at microphages and macrophages, which are scavenger cells which can attack the pathogens directly. Now we will look at natural killer cells which destroy infected or abnormal body cells. So these NK cells do not attack the pathogens directly. Instead, they destroy the infected or cancerous body cells. These were discovered in 1975. These look similar to lymphocytes but contain granules with toxic chemicals. These recognize dividing or abnormal cells without needing any specific immunity. And NK cells can kill cells by releasing cytotoxic granules inside the cells and eventually leading to their leakage and death. And it is the third most abundant lymphocyte type after B and T cells. And these NK cells develop from hematopoietic stem cells and mature in the bone marrow and liver. Now, if we talk about lymphocytes, the nature of lymphocytes, uh, lymphocytes actually enable the immune system to recognize and respond to different foreign substances. Okay, and it's usually inactive on dormant until it gets activated by specific signals. So lymphocytes are very briefly or broadly small rounded cells with a large nucleus and little cytoplasm. And lymphocytes can circulate through blood and lymph to move around the body. And humans have approximately 2 into 10 to the power 12 lymphocytes, but only nearly 1% are in the bloodstream at a time. So where are the rest? So location in the lymphatic system, most lymphocytes are actually located in lymphoid tissues. Here we can see those uh, lymphoid tissues are present in the bone marrow. Here, spleen. Here and thymus in this area. Then you have tonsils, right? In this part near our like near the throat. Then you have intestinal linings and definitely lymph nodes, which are present at various locations in the body. Okay, so there the lymphocytes are present there, and these tissues overall, all these lymphoid tissues form the lymphatic system, and lymphocytes move through these networks of lymph capillaries. This figure shows that, allowing interaction with macrophages and other immune cells. Now, if we talk about the lymphocyte types, so one is T cells, which develop from stem cells in bone marrow, but it matures in the thymus. It is developing in the bone marrow, but it matures in the thymus. Hence the name T cells. It actually T cells make up about half of all lymphocytes, and it is responsible for cell-mediated immunity. T cells directly attack infected or abnormal body cells, and it recognizes pathogens inside body cells. But also it can help in regulating the function of B cells. And when we have B cells develop and mature in the bone marrow, and it provides humoral immunity. And it produces antibodies that bind to antigens or pathogenic substances inside the body fluids, and it can also detect pathogens outside the body cells. And interaction between T and B cells. So immune responses often involve both cell mediated, which is the T cells, and humoral immunity, which is conferred by B cells. And T cells regulate and support the activity of B cells. Okay. Oh, so this is again a scanning electron micrograph of a T lymphocyte, healthy T lymphocyte. Okay. I mean T lymphocyte from a healthy person. So it hasn't yet engaged with any foreign pathogen. So recognition of foreign molecules. How are these foreign or how are these pathogenic molecules recognized recognized by these cells and the immune system? So receptor molecules, there are receptor molecules. So lymphocytes recognize foreign substances um, because they have certain specific receptor proteins, and receptors fit antigens like a key and a lock. B cells secrete antibodies which are free versions of their receptors, and T cells can only have surface receptors and they do not secrete antibodies. So if we talk about, this is the B cell which releases antibodies, okay, these yellow ones, and that can attack the pathogen. So these are viral organisms. Antibodies are these yellow ones, okay, secreted from the B cell, and these are all pathogenic bacteria or antigen, okay, viral pathogen, bacterial pathogen. So all these antibodies are actually kind of attaching to the pathogen or antigen, and then not these are B cells which are releasing antibodies, and there are all the, we talked about macrophages and stuff, right? So those specialized cells are also there which will eventually phagocytose uh, these antigens or these pathogens. Okay, so overall all these cells and different types of cells actually come together to fight against a pathogenic attack. Okay. And there are, if we talk about the antigens, antigens are foreign molecules that bind to lymphocyte receptors. Examples can be viruses, bacteria, fungi, protozoa, pollen, dust, or any transplanted tissue as well. Immunogens are antigens that trigger an immune response. Haptens are small antigens that cannot trigger an immune response unless it is attached to a larger molecule. Okay. And how are antigen antibody interactions determined? So basically there are antigenic determinants which are called epitopes, which are specific regions. There can be multiple specific regions on the foreign substance, and each epitope can be recognized by different lymphocytes, and some different substances share similar epitopes causing cross reactivity. So what are the recognition differences? If we talk about, B cells can detect antigens outside the cells because it is secreting the antibodies, whereas T cells detect antigens inside the infected body cells. If we talk about the diversity of lymphocytes, the immune system can recognize almost any complex molecule, and each lymphocyte has one unique antigen receptor. And when a lymphocyte recognizes its antigen, it multiplies. Okay. And this has a term which is called clonal expansion. How does this diversity come about? Okay. So what is the genetic basis of receptor diversity? Receptor diversity comes from rearrangement of gene segments during lymphocyte development, right? And a limited number of genes combine in so many different ways, thereby giving, how we in last tutorial session only we looked at alternate splicing, right? So different um, coding sequences, regions of the coding sequences come in different arrangements to give rise to different protein sequences, and eventually uh, as in different amino acid sequences, and eventually a different protein altogether, right? So similarly, due to gene, a limited number of genes can arrange in different ways, thereby producing different types of receptor proteins. Okay. So additional diversity occurs through junctional diversification. There are changes during gene recombination, or there can be somatic mutation where the DNA sequence itself can change during cell division. Okay. Now, if we talk about somatic mutation, it occurs in lymphocytes during immune responses. These somatic mutations produce slightly changed receptors in descendant cells, and there are some variants which can bind antigens more effectively, thereby improving immune response. Now, if we talk about the basic structure of immunoglobulin or antibody, antibodies are proteins or immunoglobulins, which are also shortly, abbreviated as Ig. So Igs belong to globulin class of proteins, and all the antibodies have this basic Y-shaped structure. You see, this is the Y-shaped structure here. This is how an antibody looks like. So if we talk about the chain and the overall structure here, each antibody consists of four polypeptide chains. This one, this two, one blue, two blue, three pink, 4 pink. Okay. Each antibody consists of 4 polypeptide chains where 2 are identical heavy chains. These blue ones are the heavy chains, and these pink ones are 2 identical light chains. And all the chains are linked by disulfide bonds. Okay. So the 4 chains together form a flexible Y-shaped molecule. Then what about the antigen binding sites? Okay. We can see that there are different antigen binding sites on the antibody. It is mainly located at the tips of the two arms of this Y shape here and here. You see these ones are the regions where the antigen can bind. Okay, these are the antigen binding sites on this antibody. Um, Again, the antigen binding site, if you look at, this is the heavy chain, this is the light chain. So both the heavy and the light chains are together making up the antigen binding site. Each antibody has at least 2 identical antigen binding sites, and these sites recognize specific epitopes or antigenic determinants on antigens, right? So we talked about epitopes, right, which are specific regions on the antigen which gets recognized by or which get recognized by the antibody. So in this scenario, if this green one, light green one is an antigen, then these regions are its epitopes, okay, which are getting recognized by the antibody. And binding, this interaction actually occurs through weak chemical bonds. Now, if we talk a little more in detail about the chains, like on the antibody, so there are variable and constant regions. Okay, so each chain has two main regions: the variable or V region, which varies between antibodies and determines the antigen specificity. These regions here are the variable regions. And then there's constant region or C, which is similar within the antibodies of the same class. These ones. Okay. So when we talk about domains, so antibody chains are folded into functional units called domains. Basically, this is a protein only, right? So how a protein folds onto itself to perform a specific function. Similarly, antibody also is a protein, right? So the different chains are folded into a particular format, giving rise to the giving rise to functional domains. Okay. So if you talk about the light chain, this one here, it has one variable domain, the V region VL, and one constant domain CL. Okay, the light chains, both the light chains. Now, if we talk about the heavy chain, the bigger one here, this one. Okay, so the heavy chain has again one variable domain VH. Okay, and there can be 3 to 4 constant regions, constant domains. Okay, CH1 to CH4. You can check this. It doesn't have CH4 in this diagram. It has CH1 to CH3. Okay. And the constant domains in the tail determine the antibody's biological functions after binding an antigen. Okay. So variable and constant domains with light and heavy chains of an antibody or immunoglobulin, folded shapes of the domains, these ones. You see the foldings here. Those are also, so there are disulfide linkages everywhere between the chains, okay, between the light and the heavy chains, between two heavy chains as well. However, if you see that in between these are the foldings of the domains, right? Shapes of the domains are actually maintained by the disulfide linkages. Okay. Then there's the hinge region, this area, this where the forking, like the fork opens up. Okay, so this is called the hinge region, and you have this tail part here. And this hinge region is actually located between CH1 and CH2 on the heavy chain. This blue one is the heavy chain. Okay, so this was CH1, this was CH1, the next one was CH2 here. So this hinge part is actually located at this region. It helps in providing flexibility to the antibody, and because it is providing flexibility, so it helps eventually to attach to the antigen in a more stronger way, like in a more strong way. and it helps cross-link antigens, forming large antigen-antibody complexes that can be easily removed by macrophages. So if you look at this part, this is the hinge region, the dotted circle of an antibody molecule opens up and closes to allow better binding between the antibody and antigenic determinants on the surface of an antigen. So this B region, hinge flexibility also facilitates the cross-linking of antigens into large antigen antibody complexes. If we talk about the classes of immunoglobulins or classes of antibiotics, there are five main classes based on the differences in their heavy chain constant regions. And structural differences between the classes give them different functions in immunity. One is IgG, which is the most abundant antibody in blood and tissue fluids. It has a basic four-chain structure with two antigen binding sites. It is the only antibody that crosses the placenta and hence provides passive immunity to the fetus. Passive immunity, I mean, we will look at it later also. But just because it came here, passive immunity is something which is not already present and it is passed on via some foreign agent. Okay. IgG is also transferred to infants through mother's milk. Then there's IgM class of antibodies. This is the first antibody produced by B cells during immune response, and it often exists as five groups. Okay, exists as pentamer, where five Y-shaped units are joined together, and hence if there are five y-shaped units, then if we look at the previous here, there are five these things. So there are two antigenic determinant sites, antigen binding sites, right, in one. So if there are five, it is a pentamer, then there are 10 antigen binding sites, and hence IgM is a very effective pathogen, as in very effective against pathogens. Okay, because it binds more effectively since it has 10 antigen binding sites. So IgM causes agglutination or clumping of microbes, especially bacteria. Then you have IgA. It is the main antibody in the secreted fluids from our body, like tears, saliva, respiratory secretions, intestinal fluids. Colostrum is the milk that is fed to a newborn baby. And then IgA is produced by B cells in mucous membranes. Usually, IgA exists as a dimer. Two IgA molecules are joined together with a special secretory protein, and this immunoglobulin is produced almost daily, but it is less stable than IgG. IgG exists as a pentamer. Okay. Then IgD, which is found mainly on the surface of developing B cells. It is present in little amounts in the bloodstream, and it is speculated to be involved in B cell activation, although its exact function is a little unclear. Then you have IgE, which is present in very low concentrations in the blood, and it binds to basophils and mast cells. And when antigens bind, these cells release histamine and other chemicals, causing allergic reactions. And IgE also helps in defending against parasitic infections. So in general, if we talk about the normal antibody production, the body maintains an optimum antibody level by continuously producing and breaking down uh, like not only the antibodies, eventually the signaling pathways and other regulatory factors, which leads to the production of antibodies. Okay. If we talk about the five antibody classes, immunoglobulin classes, the relative amounts in blood, IgG is present in the maximum amount, followed by IgA, then comes IgM and IgD, and IgE immunoglobulin is actually present in very low quantities. Even without infection, B cells produce a small baseline level of antibodies. Okay. Now, when we talk about antibody class switching, B cells first produce IgM when activated by an antigen. Later, they may switch to producing IgG, IgA, or IgE. And then there are memory B cells which quickly produce IgG or IgA during repeat infections. Okay. And the type of antibody produced depends on the type of antigen, location of the infection, and the cytokines released by T cells. Now, if we talk about cell-mediated immune mechanisms, T cells play an important role in immunity separate from antibody-producing cells, and cell-mediated immunity protects against pathogens that escape antibody defenses, especially viruses and intracellular parasites. Many viruses spread directly from cell to cell, avoiding antibodies in the bloodstream. People who can't really produce antibodies are highly susceptible to bacterial infections but less vulnerable to viral infections due to cell mediated immunity. And then this immune response destroys the infected body cells, preventing microbes from reproducing and exposing them to other immune defenses. The mechanisms of cell mediated immunity, helper T cell response. Activated helper T cells release cytokines that regulate immune responses. A key cytokine is gamma interferon. We looked at interferons, right? Alpha, beta, and gamma produced by different types of cells. And gamma interferon it increases the ability of macrophages to kill microbes they ingest, stimulates natural killer cells to enhance immune defense. Then there are cytotoxic T cell response by T cells, which are called killer T cells, which directly attack the infected cells. The killer T cells bind to target cells, displaying specific antigens on their surface. And once they attach, they damage or kill the infected cells. So, what are the different methods cytotoxic T cells use to kill the target cells? One is the release of cytotoxic molecules. Cytotoxic T cells release pore forming proteins, which are also called as perforins, and these proteins create holes in the target cell membrane. The cell membrane becomes permeable, causing leakage of cell contents and cell death. Also, it induces apoptosis, which is called programmed cell death. So there are some cytotoxic T cells which trigger apoptosis in infected cells or target cells. This often occurs through the activation of this particular protein, Fas protein, on the target cell surface. An interaction with these T cell proteins and Fas protein on the surface of the like target cell or infected cell sends a signal to the cell nucleus. The infected cell then can self-destruct, destroying the virus inside it. Okay. Now, if we talk about immunization techniques, I mean methods, one is the prophylactic immunization. Prophylactic immunization is the artificial creation of immunity to prevent infectious diseases, right? So it can be of two types: passive immunization and active immunization. Passive immunization, it actually helps, it gives immediate but temporary protection using preformed antibodies from another individual. This is used when infection or toxins can act too quickly for the body to respond. For example, in case of snake bites, rabies exposure, tetanus, or botulism, passive immunization is the first method. Okay, first line of kind of defense, and mainly IgG antibodies are given as anti-serum, and protection usually lasts for 2 to 3 months. And when we talk about active immunization, it actually stimulates the body to produce its own antibodies and memory cells via vaccination. So the living organism either killed or decapitated uh, or weakened, those pathogens are introduced inside the body, and the body itself produces its own antibodies and memory cells, okay, against the injected pathogen. So it can again be of different types, like toxoid vaccines, which are inactivated toxins. Then there are killed vaccines, which are dead microbes, which are introduced inside the body. Then there are attenuated vaccines, which are weakened live microbes. And then there are recombinant vaccines, which are genetically engineered microbes, which produce the specific antigen against which we need to produce the antibodies. So vaccines are usually injected, though some can be oral or mucosal. Okay. So the importance of immunization: vaccination is highly effective and cost efficient in preventing diseases. Mass vaccination has nearly eliminated diseases like diphtheria, polio, and measles. High vaccination coverage can lead to disease eradication, as seen with smallpox. Now, if we talk about the development of immunity in major animal groups, The evolution of the immune system cannot be traced through fossils, so it is studied by comparing immune abilities of living animals. And most animals can actually recognize self and reject foreign substances or non self substances, indicating some form of immunity. Now, if we talk about immunity in invertebrates, even simple organisms can distinguish self from non self. Examples, sponges can accept grafts. Sponges can regenerate, right? We looked at like porphyrins. So sponges accept grafts from their own colony but reject those from others. There are higher invertebrates like worms and starfish, which can destroy foreign tissue using phagocytic cells and lymphocyte-like cells. Then there are insects which can phagocytose to destroy the pathogens. They have substances in their body fluids that can cause foreign cells to clump, which eventually lead to their destruction. And insects can actually develop limited immunity to certain infections. Now, if we talk about vertebrates, vertebrates have the most advanced immune systems. Lymphocytes and antibodies appear only in vertebrates. If we talk about the early vertebrates like jawless fishes, lampreys, hagfish, they have weak immune responses and they lack major lymphoid organs like the thymus and spleen. However, if we move towards the advanced fishes like the cartilaginous and bony fishes, these have thymus and spleen and can produce IgM antibodies. However, they lack the lymph nodes, but they have the lymphocyte clusters in their guts. Okay. Now, coming to the terrestrial vertebrates, amphibians, reptiles, birds, and mammals possess a fully developed immune system, thymus, spleen, bone marrow, and lymph nodes, and can produce IgM and IgG antibodies. IgA antibodies appear in birds and mammals. IgE antibodies are found only in mammals. Now, if we talk about the complement system, the evolution of this system, the complement system or the immune proteins likely evolved faster than antibodies. If we talk about the primitive fishes, they have been found to have partial complement systems while higher vertebrates have complete systems. The complement system has been highly conserved in evolution, showing its important role in immunity. And types of antibodies, if we talk about, there can be monoclonal or polyclonal antibodies. Polyclonal antibodies or PABs are produced by multiple B cell clones and it can actually recognize multiple regions of the same antigen or multiple epitopes of the same antigen. So, how is it produced? The antigen is injected into the animals. Whatever immune response is uh, kind of what sparked, then the antibodies are collected from the serum. So, advantage of polyclonal antibodies, it's a cheap and quick way to produce antibodies, which is done within 3 months. It has high sensitivity and strong overall binding. It is good for detecting low abundance proteins and native proteins. However, each batch of antibodies secreted by the B cell clones can have variations and there's higher risk of cross reactivity. Now, if we talk about monoclonal antibodies or MABs, these are produced from identical B cell clones or a single parent cell, and it can bind to only one specific epitope or one specific region on the antigen. Okay. So, how is it produced? It is actually produced via something called hybridoma technology, where antigen is injected into the mouse, spleen B cells are isolated, fused with uh, constantly dividing myeloma cells. Then there's hybridoma that produce identical antibodies. So we'll look at hybridoma in a little more detail in the next slide. Advantages, high specificity and low cross-reactivity, consistent batches and large-scale production, good for diagnostics and therapeutic drugs. The disadvantages are expensive and slower to produce. Binding may change when labeled. Key idea, polyclonal antibodies have multiple epitopes, like can bind to multiple regions of the antigen, and it's more sensitive and cheaper. However, polyclonal antibodies have a single epitope and highly specific, and it is consistent for diagnostics and therapy. Now, when we talk about hybridoma technology, it is a technique to produce monoclonal antibodies by fusing antibody producing B cells with the immortal myeloma cells, enabling continuous specific antibody production. This was actually developed in 1965 by Kohler and Milstein, and they were awarded the Nobel Prize in Physiology. And the principle basically is, first, you have to immunize the animal, usually mice, which are immunized with the target antigen to stimulate B cell production. Then these B cells are fused with myeloma cells using PEG or electrofusion, thereby forming hybridomas with antibody specificity and immortality. Then you eventually select. These hybridomas are cultured in HAT medium. HAT medium contains hypoxanthine, aminopterin, and thymidine, which selectively allows only fused cells to survive. However, unfused B cells can die naturally, and myeloma cells cannot survive in HAT medium because these myeloma cells lack HGPRT enzyme. So we look at the enzymes and stuff in the next slide. And then eventually, once you select the hybridoma by culturing the cells in HAT medium, then hybridomas producing desired antibody are selected and cloned via limited dilution or FACS to ensure monoclonality. Then expansion and preservation. Selected hybridomas are expanded and cryopreserved for long-term use. So what is the advantage? High specificity, definitely, because it recognizes a single epitope and there's minimal cross-reactivity. Reproducibility, it is consistent and renewable unlike polyclonal antibodies. Versatility, used in diagnostics, therapeutics, research, and immunotherapy. What can be the applications of hybridoma technology? Medical diagnostics, which helps to detect pathogens, biomarkers, disease antigens. Therapeutics, treat cancers, autoimmune diseases, and transplant rejection. It can be used in research as well for protein identification, purification, functional studies. And what can be the limitation is that it is time consuming because immunization, fusion, and screening can take much longer. Species specificity, mouse derived antibodies can trigger immune responses, which is mitigated by chimeric or humanized antibodies. And definitely because the overall technology involves a lot of efforts as well as resources, so these are costly, like really costly, and because it requires special equipment and expertise. So this is what is happening. Antigen is injected inside mouse. Immune cells, the B cells that are produced are isolated. So these are basically the antigen for antibody forming cells against this specific antigen that has been injected. Then there's tumor cell, or myeloma cell, which is immortal. So overall, once this is fused, then you finally have a continuous supply of antibody producing cells, producing antibodies against the specific antigen. Then hybridomas are screened for production of desired antibody, and antibody producing hybridomas are cloned. Eventually, this gives rise to monoclonal antibodies. So we were talking about HGPRT, why myeloma cells and stuff cannot grow on HAT medium. Okay. So nucleic acid synthesis is essential for growth and multiplication. HAT medium inhibits the de novo pathways, and cells with salvage pathways containing HGPRT enzyme can survive. Okay. But myeloma cells depend solely on the de novo pathways for nucleic acid synthesis, whereas kidney cells has both de novo and salvage pathways. So in the presence of this particular medium, individual myeloma cells or kidney cells will not survive, whereas only the hybrid cells are able to survive. Now, when we talk about agglutination reactions, agglutination is the process of linking together antigens and antibodies and forming visible aggregates. Agglutination reactions involve particular antigens like bacteria, virus, or RBCs, and these are very sensitive and reproducible. But these reactions can also be divided further into direct and indirect agglutination reactions. If we talk about direct agglutination reactions, it is used in test diagnostics for a large number of cellular antigens like RBCs, bacteria, and fungi. For blood group determination, also this is used. So this test is carried out in plastic microtiter plates that have several shallow wells. Formation of antibody antigen mat, which sinks to the bottom of the well, is an indication of the antigen being present inside the body. However, in the negative reaction, agglutination does not occur and insufficient antibodies are present to cause the linking of antigens. Okay. So, if we talk about blood group typing, blood typing is accomplished by mixing drops of blood with sera containing antibodies against the ABO and the RH systems. Okay. Now, if the blood cells clump, the antibody has bound to the appropriate antigen on the cells, and the blood type matches whatever antibody caused agglutination. So, if our blood agglutinates in anti-B tube, then we have the B antigen and we have blood group type B. And blood types are indicated by both the ABO and RH antigens present. So here, what is the antibody? Anti-A, that means um, agglutination has happened, and that means blood group is A. If there is anti-B, agglutination has not happened, that is also an indication that it is blood group A. Similarly, if we are providing anti-A antibodies and there is no agglutination, that means A is not present. Uh, the other one is B is present, right? So if anti-B is given, then coagulation will happen and which will suggest that the blood group type is B. So basically, all these anti-A, anti-B are basically antibodies designed against specific substances or molecules or chemicals present on the surface of individuals having, like, RBCs of individuals having blood group A and B. Okay. Now, if anti-A also leads to coagulation or agglutination as well as anti-B, then it is an AB blood group type. Whereas for both anti-A and anti-B, there is no agglutination, then the blood group is O. Similarly, there is another antibody which is anti-D. If it's it should be positive. So then it is RH positive. If this is negative, it is RH negative. Okay. And then there are indirect agglutination reactions, which is adaptation of soluble antigen for agglutination reactions. This is again a type of diagnostic test which is rapid and used for detecting soluble antigens like streptococcus bacterial agents. If the antigens are adsorbed onto particles like RBCs or beads, soluble antigens can respond to agglutination test. So the antibody reacts with the soluble antigen adhering to the particles. Therefore, the particles agglutinate with each other as these do not do in the direct agglutination tests. We talk about the vesicular transport. What are the organelles mainly present in vesicular trafficking? It's mainly the ER, the Golgi apparatus, and lysosomes. Okay. So the main function of these organelles is to manage the distribution of material, food particles or proteins throughout the cells. There are three different organelles: ER, Golgi apparatus and lysosome, which coordinately work together to maintain vesicular transport of material across the cell. Eukaryotic cells take up the solid material from outside the cells through a process called endocytosis, whereas uptake of liquid is through a process called pinocytosis. Similarly, material is secreted out of the cells through exocytosis. In addition, intravesicular system delivers protein synthesized in ER to different organelles. During endocytosis, the material present outside the cells binds to the cell surface through cell surface receptor and are trapped inside a membranous structure called the endosome. Okay, this one is the endosome here. Okay, this one. When it is bound by a membranous structure, it is endosome. And endosomal vesicles are fused with lysosome to form the late endosome. This one here, this lysosome red one fuses with this early endosome to form the late endosome. Okay. And in the late endosome, with the help of lysosomal enzymes, the material or the pathogen is digested, and endosome is further fused with the Golgi bodies here. This one, this fusion is taking place and delivers the content for further distribution. In the similar manner, during secretion, vesicles originate from Golgi bodies and fused with the plasma membrane to release the content outside the cells. Okay. Of course, there are Golgi bodies, which are the functions are protein sorting, protein modifications, and proteolysis. So we read about individual organelles in much depth in earlier tutorial sessions as well. So if we talk about the cis phase, each lamellae here, cis phase receives material or vesicles from the endoplasmic reticulum. Then there's medial Golgi or medial cisternae is the actual place where proteins are covalently modified with sugar. And then there's trans Golgi, which faces towards the plasma membrane. And this site eventually sorts the vesicle for their for their destination. Okay. Then there are lysosomes, which are suicidal bags. They are due to their role in autophagy, where the lumen is extremely acidic. It contains different kinds of enzymes like proteases and cytolytic enzymes to degrade the ingested pathogen. Function is to degrade the ingested food material for delivery through the vesicular system, and degradation of pathogenic bacteria, degradation of some old proteins. So if we talk about the trafficking signal here, glycosylation is a dire function of biosynthetic secretory pathway in the ER and Golgi apparatus. Approximately 50% proteins characteristically expressed in a cell go through this alteration, which involves the covalent addition of sugar moieties to specific amino acids. Mostly soluble and membrane-bound proteins expressed in the ER undergo glycosylation, including all the secreted proteins, surface receptors, and ligands. Moreover, some proteins that are transferred from the Golgi to the cytoplasm are also glycosylated. Okay. So now let's look at some of the questions. So which of the following best describes hybridoma technology? Fusion of T cells with macrophages to produce antibodies? No. Fusion of B cells, which is antibody producing, with myeloma cells, which divides continuously to produce monoclonal antibodies. This is the correct option. Fusion of B cells with fibroblasts? No. Fusion of dendritic cells with T cells? No. Correct option is option B. Okay. Why is HAT medium used in hybridoma selection? Second question. Because it allows only fused hybridomas to survive. Okay, because it does not allow myeloma cells which do not have the HGPRT enzyme, and it enhances antibody production. No, it allows only B cells to survive. Which of the following is a major advantage of monoclonal antibodies over polyclonal antibodies? Monoclonal recognizes a specific region, specific epitope, so it does not really recognize multiple epitopes. This is polyclonal antibodies. Lower cost of production is for polyclonal antibodies, not for monoclonal antibodies. This is much more complicated and requires specific expertise. Monoclonal antibodies have high specificity to single epitope. This is the correct option, option C. Okay. The first step in hybridoma technology involves the fusion of myeloma and B cells, immunization of a host animal with antigen, cloning hybridomas by limiting dilution, cryopreservation of hybridoma cells. So the first step is basically to immunize, to inject the antigen inside the animal to produce B cells, which will eventually produce antibodies against that antigen. So correct option will be option B. After the B cell producing the specific antibodies are cultured, then that B cells will eventually be fused with myeloma cells. Okay. But the very first thing is immunization of the host animal with the antigen. Which of the following statements is correct regarding hybridomas? They can proliferate indefinitely and produce specific antibodies. This is the correct one. They produce polyclonal antibodies. No. They produce monoclonal antibodies. They are derived from T cells. No. They are derived from B cells, and they are cryopreserved. Okay. It's not true that they cannot be cryopreserved. Correct option is option A. Now, hybridoma cells are immortal and can continuously produce monoclonal antibodies. Myeloma cells fused with B cells confer the ability to divide indefinitely. Okay. So both these are actually true, and this reason is the correct explanation of the first statement. Okay. So next is the first statement is monoclonal antibodies are generated by hybridomas. it can recognize multiple epitopes on the same antigen. This is false. And each hybridoma is derived from a single B cell cloning producing a single type of antibody. Okay, monoclonal antibodies generated by hybridomas. So different hybridomas. If we here, they like hybridomas are talked about from multiple hybridomas are talked about. It's not talking about each hybridoma. So each hybridoma can recognize specific epitope on one antigen. But if multiple hybridomas are produced, where the B cell clone, one B cell clone is produced, as in is producing antibodies against another epitope of the same antigen, that is possible. Okay. So this is fine, but each hybridoma is derived from a single B cell clone producing a single type of antibody. So this is R is true, each hybridoma. And here, monoclonal antibodies generated by hybridomas can recognize multiple epitopes on the same antigen, which is false, right? And so the correct option is A is false, R is true. Which of the following is true about T lymphocytes? They produce antibodies directly, they mature in the bone marrow. Uh, so they produce antibodies directly, they mature in the bone marrow, they mediate cell-mediated immunity, they are part of the innate immune system. So the correct option is cell-mediated immunity because they do not produce antibodies directly, and even T cells help in, kind of protecting against pathogens inside the cells. They mature in the bone marrow. No, they are produced in the bone marrow and they mature in the thymus. And definitely they are not part of the innate immune system. They are part of the adaptive immune system. Which part of an antibody determines its antigen specificity? So the constant region of heavy chain? No. If you remember the Y-shaped structure there, the variable regions of the light and heavy chains determine the antigen specificity. So correct option is option B. The flexibility of the hinge region helps in the binding interactions, but not really specifying the and like interactions against, interactions of the antibody against the antigen, but it provides the flexibility to bind to the antigen more tightly or weakly. Okay. And then which of the following correctly pairs antibody class with its function? IgG is primary antibody in first exposure. IgM crosses placenta to protect fetus. IgE mediates allergic reactions. IgD main secretory antibody in milk. Correct option is this one. IgE actually mediates allergic reactions. The others are false. Okay. Um, so which of the following statements about antigens is correct? All antigens are immunogens. Haptens are immunogenic by themselves. Epitope refers to the part of the antigen recognized by a receptor. Antigens are only found on pathogens. So all antigens are immunogens. Haptens are immunogenic by themselves? No. Haptens are like small antigenic molecules, but when these are bound to a larger molecule, then they can be made immunogenic. All antigens need not be immunogens. An epitope refers to the part of the antigen recognized by a receptor, which is fine. Antigens are only found on pathogens, not necessary. So the correct option is option C. Okay. Which is what the specific, it is talking about the specific region that gets recognized by a receptor. Next is again statement and assertion and reasoning type of question, where the first statement is IgM is the first antibody produced during an initial immune response, and IgM exists as a pentamer providing high avidity for antigens. Avidity is basically uh, refers to the how tight the interactions are between the antigen and the antibody. Okay. So high avidity means the interactions and the specific recognition of the antigen by the antibody is really strong. Okay. So the first statement is IgM is the first antibody produced during an initial immune response, and next is IgM exists as a pentamer providing high avidity for antigens. Okay. So the options are like both these statements are true, and the second one explains the first statement. not this one, and oh, sorry, I already provided the answer. So both these statements are actually true, and because IgM is the first antibody produced during an initial immune response. Why? Because it exists as a pentamer, so it has two, like each Y shape has two antigen recognizing regions, right? So um, if it exists as a pentamer, then it has 10 antigen recognizing regions, right? The antibody has 10 antigen recognizing regions, so it will be a stronger interaction as well as stronger recognition and tighter recognition by this particular antibody, which provides high avidity or higher and tighter recognition for the pathogens or foreign substance. Hence, this is the first one to be produced during an initial immune response. So, both these statements are actually correct, and also the second one explains the first one nicely. Okay. The next one is the hinge region of an antibody molecule provides flexibility for antigen binding. The next one is the hinge region is formed by variable domains of heavy and light chains. The first one is correct, definitely. It gives flexibility for antigen binding. And hinge is formed by variable domains of heavy and light chains. Variable domains are actually at the top one. Next comes the lighter chains, right? And then um, as in the constant regions of the light and heavy chains. So then comes the hinge region. So variable domains are not really involved. So the correct answer is option C, where the first statement is correct and the second statement is false. Okay. So yes, that's it for today's session. We will meet again the next Sunday with week 8's week 8's lecture content. We will summarize that and look at few questions based on the content of week 8. All right, we'll stop sharing the screen now. Um, Thank you for joining, and I will end the meeting now. Okay, see you next Sunday.